This is the introduction to the neural systems class. So in this presentation I will just talk about the course content, and then I will talk about brains and their constituents, the neurons, the glia, and then visualize neurons and their compartments. And for the biological part of the class I will follow this book over here, Neuroscience by David Purves, which is a very nice book, can highly recommend it. It's a bit heavy, so that's why I cut it into pieces, so I don't have to carry around the book all the time. Okay, so the lecture covers essentially biology for one half. So we cover neurobiology and neuroethology. Neurobiology is the study of biological processes encapsulated in neurons, and neuroethology is the study of natural behavior and its neural mechanisms. So I guess for the biologists, many of the topics in biology here will be repetitions of known facts. So we'll cover the electric signals of neurons, active conductances, animal experimentation and ethics, then passive membrane properties, synaptic transmission and potentials, synapses and synaptic plasticity, and so much about the biophysics. And then neuromodulators, brain architectures and special examples, birdsong, vision, classical conditioning. And so this forms the biological part of the class that will mainly cover the first half of the semester, and then the second half will deal with computation, so the information processing that is carried out in the brain. And we study the computation either using estimation theory or we model it using methods in computational neuroscience. So in terms of computation, we look at neural rate equations and neural networks, and principal component analysis, just to get us warmed up for many of the things that are to come, like receptive field models, sparse coding, memory models, Hopfield networks, population encoding, estimation theory, Bayesian estimation, and causal inference and reinforcement learning. This is not complete as the topics that we'll cover in computation. So sometimes the computation will be quite advanced. We look at complex equations, and so for those of you who are not so familiar with analysis and algebra, it may be good then to catch up on some of these topics. I don't have a nice book to hand out for these computational aspects of the class, unfortunately. So what are neural systems? A neural system, I would say, can be seen as just the combination of a behaving agent, a behaving animal, and a brain. And the brain by itself is boring, I find, and the best way to start to look at the brain is to first look at the behavior, because that's what brains do, right? Neural systems are composed of a body with sensors and actuators, which are the eyes, the ears, the skin, and the actuators are almost always the muscles. So that's what the brain needs to control, right? The goal of neural systems is to produce behavior. To understand neural systems, we must research the behavioral algorithms and their implementations in terms of the brain's neurobiology. So the first lesson here is that when we look and carefully look at an animal and we study how it solves a problem, we learn about the algorithms that the brain implements. But how is it implemented? It's not like in a computer with digital processing units. It's implemented in the neurobiology of the brain. So the brain gives us the implementation of the algorithm. And these two views will keep elaborating in this class. Let's look at some neural systems. So what is really remarkable about the neural systems is that they come from essentially one cell, right? The development of the nervous system is one of the most amazing processes on Earth. From only the set of genes and essentially a single cell, a walking bird can hatch from an egg. And this process of self-assembly is far from understood, and much research needs to be done to achieve a better understanding. Right? It's really something we are far from being able to mimic in engineering, this kind of self-construction that is going on in an egg, for example. So all the information about the body, the brain, and the basic behavior is contained in the DNA and the laws of molecular biology. Yet the number of genes does not seem to be the critical factor for determining the complexity of an organism. For example, the potato has 60% more genes than a human, and yet we think we are much more complex than a potato. Maybe the number of neurons gets us closer to reflecting the level of intelligence of an organism. C. elegans has about 100 neurons, it's 302 to be precise, it's this worm over here. And then the fruit fly has about 100,000 neurons, the mouse has 100 million neurons, and the human maybe 100 billion neurons, right? So we clearly have more neurons than any of these other animals, but we are not the champion, right? Some whales have twice as many neurons as humans do, and it is unclear what this means for assessing their intelligence. Let's look at brain size. So here's a comparison of brain size, roughly in proportion to the true size. So the heaviest brain is that of the sperm whale. It weighs about 8 kilograms. So humans have a lighter brain, right? Ours weighs maybe 1-2 kilograms. So we stand out in terms of the size of the brain relative to the body weight, because the ratio of brain weight to body weight is about 1 to 5,000 in a fish. It's about 1 to 500 in reptiles, and then 1 to 220 in birds, 1 in 180 in mammals, and then 1 in 50 in humans. So in other words, our brain weighs about 2% of our body, which is very little. But it consumes about 20% of the energy. So it is definitely important, and the energy we consume is devoted to the brain. So let's look at the neuron compartments. What are the main compartments? So this class will not be much about molecular biology, but we need to just get our heads around some words over here. So let's look at some important compartments. There are the mitochondria. They are here, there. Mitochondria are the energy factories of the cells. The energy currency for the work that animals do is the energy-rich molecule adenosine triphosphate, ATP. So ATP is produced in the mitochondria. These are the factories. And then ribosomes. They are here. Ribosomes is a minute particle consisting of RNA and associated proteins of living cells. Ribosomes bind messenger RNA and transfer RNA to synthesize polypeptides and proteins. So they're very important. These are really the factories where the proteins are made that power everything we do and think. Then the nucleus over here contains the majority of the cell's genetic material. This is obviously DNA molecules along with a variety of proteins that form chromosomes. Then the Golgi apparatus over here is a complex of vesicles and folded membranes within the cytoplasm. It is involved in secretion and in intracellular transport. So the Golgi apparatus is used to transport those proteins that are made in the ribosomes to their destination. Then the endoplasmatic reticulum. Over here is a network of membranous tubules within the cytoplasm of eukaryotic cells, which means all cells that have a nucleus. It is actually continuous with the nuclear membrane, so it's just surrounding here the membrane of the nucleus. It usually has ribosomes attached to it and is, of course, involved in protein synthesis and in lipid synthesis. And here, the more familiar compartments of neurons are the dendrites over here. These are the dendrites, these appendages here. It's an extension of a nerve cell along which impulses received from other cells are transmitted to the cell body. So the dendrites, they are the places where the neuron gets the inputs from other neurons. And the outputs is signaled along the axon over here, in blue over here. So the axon is the long thread-like part of a nerve cell along which impulses are conducted from the cell body to other cells. Then last here on this picture we have myelin. This is this thing over here, also here, covering the axon. It's a fatty white substance that surrounds the axon of some nerve cells, forming an electrically insulating layer. So this is already more or less all the subcellular compartments that we're going to see in this class, except of course at the synapses where we'll see many things. But before we get there, let's look at modern neuroscience. So neuroscience, modern neuroscience is about only 150 years old. So it's kind of a young science if you compare it, for example, to physics. It has started with the first descriptions of brain anatomy by Camillo Golgi and Santiago Ramón y Cajal. So these are the two heroes of modern neuroscience. Golgi invented the method of silver salts that sparsely labels neurons. So if the staining was more dense, right, if these salts that he put on fixed brains, if the staining was more dense, then the entire tissue would turn black and we wouldn't see anything. So it's thanks to the inefficient labeling of these silver salts that he was the first person to see that the brain is composed of these tree-like structures. And he correctly recognized cell bodies, dendrites and axons. So Golgi was a proponent of the reticular theory, hypothesizing that the brain is a dense network of singular entity, the reticulum or net. So it was not clear at that time yet that the brain was not composed of individual units, the neurons. Cajal, by contrast, he thought that the brain is composed of these individual neurons. This is called the neuron doctrine. So there was a kind of intellectual fight going on between these two camps. So here we see really some images from Golgi, silver salts. Here too, this is a Purkinje cell. And there's other ways of visualizing neurons here with fluorescent dyes. You can intracellularly inject an enzyme with a thin pipette. Enzymes are proteins that act as catalysts and help complex reactions occur. You can also stain here cell bodies, only the cell bodies, using crescent violet, and Nissl stain. You can stain entire brain sections using Nissl stain, which just visualizes the nucleus of neurons. So much is known about the structural features of neurons thanks to electron microscopy. So the work of Charles Sherrington in the 1950s with his electron microscope, he led to the discovery of the synapse. The synapse is a junction between two nerve cells, right, consisting of a minute gap across which impulses pass by diffusion. So here we see a synapse over here. This whole thing is a synapse. So Charles Sherrington's work essentially settled the debate, right, in favor of the neuron doctrine. We all know now today that the brain is composed of many neurons. And so he was visualizing, for example, here, this thing in black here, it's the postsynaptic density. These are receptor molecules. These things over here, these little spheres are vesicles, right? So the postsynaptic density is a protein-dense specialization attached to the postsynaptic membrane. And so these appear in microscopic imagery as electron-dense regions, and synaptic vesicles store various neurotransmitters that are released at the synapse. Right? The release of the neurotransmitters is regulated by a voltage-dependent calcium channel, as we're going to see. And vesicles are essential for propagating nerve impulses between neurons and are constantly recreated by the cell. And the main neurotransmitter types you're going to see: excitatory transmitters, glutamate, inhibitory transmitters, GABA, and others. And this thick insulating layer here is the myelin that also appears as electron-dense material. Today, we use genetic engineering to visualize neurons, right? And not only neurons, and a plethora of cell types and cellular compartments, right? For example, a popular method is that viruses can be engineered to transfect a cell or group of cells to express a fluorescent protein of some type, right? So cell and protein type are encoded by the promoter and the reporter gene that are inserted into the viral DNA, right? A promoter is a region of DNA that initiates transcription of a particular gene. A promoter, right, is essentially turns the gene transcription on. It's like the on switch, and the promoters are located near the transcription start site of genes on the same strand and upstream on the DNA. And the reporter gene, right, often simply reported as named as the reporter, is a gene that researchers attach to a regulatory sequence of another gene of interest. The reporter is only expressed in those cells that express the gene. So certain genes are chosen as reporters because the characteristics they confer are easily identified and measured, or because they are selectable markers. For example, a green fluorescent protein is a useful reporter gene because it turns the structure of interest green. Right? In this example over here, a peripheral axon over here is visualized. It's turned green, including the axon terminals in the spinal cord. Right? We can use several colors. Right here is just the green axon is labeled, but we can use multicolor imaging. So fluorescent labels can be attached to almost any kind of cell compartment, as shown here for the cytoskeletal protein tubulin, right? In green, sorry. Tubulin is here in green. And in A, so here the axon of a neuron is shown in red, right? And it's labeled by the microtubule binding protein tau. There's one particular protein here that turns it red. And tubulin here is a protein that polymerizes into microtubules. So that's used to label the cytoskeleton. So in B over here we see also the tips of a neuron in red, right? This is actin. Actin is a protein important for mobility and contraction, right? So in E is another visualization of a neuron cell body, and in F again the dendrite in green with again actin in red, right? So kind of these images provide a nice visualization of the more rigid parts of a neuron in green and the dynamically moving parts in red. So we can, from just looking at this still image, imagine that these red things are moving around constantly as the neurons are probing for making new contacts with new partners or getting rid of the old contacts. So neurons are not the only important cell types in the brain. The other main players are the glial cells, and I've here shown 3 images of glial cells. So neuroglia, or simply glia, come from the Greek word glue, are non-neuronal cells. So the glia are not really electrical cells. They're not really transmitting electric signals. So they are cells that maintain the homeostasis in the brain, like these guys, astrocytes. Or they form myelin, or they provide support and protection of neurons in the central and peripheral nervous system. So glial cells include oligodendrocytes, astrocytes as here, and then microglial cells. And also Schwann cells and satellite cells that are not shown here. So the human brain contains about also 100 billion glia, so the same number of glial cells as there are neurons. So astrocytes over here, they are characteristic star-shaped glial cells, so 20 or 40% of glial cells are astrocytes. They provide biochemical support of endothelial cells and form the blood-brain barrier. And they provide the nutrients to the nervous system, and they maintain the extracellular ion balance, and they repair and scar of the brain and spinal cord following traumatic injuries. Then oligodendrocytes produce the myelin sheath that insulates the neurons, the neuronal axons, right? So they're analogous to Schwann cells in the peripheral nervous system. So these guys that make the neurons covered in myelin, which then increases the conduction speed. And the microglial cells, these are the smallest, they are the brain's macrophage cells. So they act as first aid and they form an active immune defense system of the central nervous system. So these are the ones that come to the rescue when there's an injury of the brain. So this concludes this first introduction. We've seen now what the lecture is about, what the function of the brain is to produce behavior, and we've seen the main parts of neurons and also the other cells, the glial cells in the brain. And the first question we could discuss are, what are the advantages, disadvantages of light and electron microscopy for visualizing neurons? Thanks, I hope you enjoyed this lecture.